Glycolysis. The prefix glyco is referring to glucose, and the suffix lysis means breaking down. So glycolysis literally means breaking down of glucose. Glycolysis is an extremely important metabolic pathway for the breakdown of carbohydrates and production of energy. It's also an extremely ubiquitous metabolic pathway because a host of different organisms can all perform glycolysis. If you eat carbohydrates, your body is going to perform glycolysis in order to digest this and harvest energy from it. But an organism as drastically different from you as yeast can also perform this same metabolic pathway. Bakers and brewers have been exploiting glycolysis in yeast for centuries to make things like bread and alcoholic beverages. Glycolysis is a series of chemical reactions. It starts with glucose and it ends with 2 molecules of pyruvate, all while harvesting as much energy as possible. So for that reason, other compounds are involved that are here just to capture that energy that's produced. I am going to write all 10 steps of glycolysis out on the board. It is going to be packed full of details, so this is a part of the video that you are probably going to need to watch multiple times. I strongly encourage you to make a table and fill it out to help you organize the details. For each of the 10 steps, keep track of names and structures of major products and reactants, names of side molecules, the type of each reaction, the name of the enzyme that catalyzes each reaction, if reactions are reversible or irreversible, and the energy that is produced or consumed in each step. Let's get started. Step 1 of glycolysis is an inorganic phosphate transfer. So we're starting with glucose, which is a 6-membered ring containing a total of 6 carbons. And we need some source of phosphorus. So we are also going to need to use ATP in step 1. ATP is adenosine triphosphate. One of those phosphate groups is going to be broken off, so it's going to become adenosine diphosphate, ADP. The enzyme that catalyzes this reaction is called hexokinase, and this is an irreversible reaction. The product of this reaction is glucose 6-phosphate. Basically, we are adding that phosphate group onto carbon number 6. So an inorganic phosphate group moved from ATP to glucose, so we now have glucose 6-phosphate. This enzyme in step 1, hexokinase, is inhibited by the product, glucose 6-phosphate. This means that there's essentially a bottleneck on this step. We don't want to make too much glucose 6-phosphate because this is an irreversible reaction. So if the concentration of glucose 6-phosphate gets too high, this step is going to slow down. Reaction number 2 is an isomerization reaction. So remember that isomers are molecules with all the same atoms that are just bonded differently. So we are basically just going to rearrange glucose 6-phosphate and turn it into fructose 6-phosphate. So this is catalyzed by an enzyme called glucose 6-phosphate isomerase. Literally the name of the reactant, then isomerase, the type of reaction. This is a reversible step, so you are seeing the 2 directional arrows. And our product is fructose 6-phosphate. This is a 5-membered ring, but we still have a total of 6 carbons. Step 3 is another inorganic phosphate transfer. This one is catalyzed by an enzyme called phosphofructokinase 1. And much like step 1, it's an irreversible reaction, and we need a source of inorganic phosphate. So once again, we are going to be using ATP, adenosine triphosphate, and breaking one of those phosphate groups off of it so that it becomes adenosine diphosphate, ADP. And our compound is going to get that phosphate group added to it. So what we're going to end up with is fructose 1,6-biphosphate. So here we still have our 5-membered fructose ring with a total of 6 carbons, but we now have 2 phosphate groups. One of them is on carbon number 1, the other is on carbon number 6. At this point, you might be understandably confused. You probably learned a long time ago that mitochondria is the powerhouse of the cell, and the cellular energy that mitochondria makes is ATP, adenosine triphosphate. And so far in glycolysis, we have used ATP. We are using energy. You have not seen us create any energy. So pay close attention to ATP and ADP throughout the glycolysis pathway. Think of ATP as our currency for energy. Step 4 is a carbon 3 to carbon 4 bond cleavage. We are actually going to be breaking our compound in half. This is catalyzed by an enzyme called aldase, and it is once again an irreversible reaction. But because we're breaking our compound in half, we now have 2 products. One is dihydroxyacetone phosphate, and the other is glyceraldehyde 3-phosphate. Notice that these 2 compounds are isomers of each other. That is important for step number 5. Step 5 is an isomerization reaction. This is catalyzed by an enzyme called triose phosphate isomerase. And in this step, there's a rapid interconversion between the 2 products of the last step, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Importantly, glyceraldehyde 3-phosphate is what can continue on to the next step. Step 6 is an oxidation and phosphorylation reaction. So this is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. And as the name suggests, the reactant is going to be the glyceraldehyde 3-phosphate. This is a reversible step, so we're getting the 2 directional arrows. We also have a couple of other molecules that we care about. So we need the coenzyme NAD+ as a reactant, and we need some inorganic phosphate. We're going to spit out NADH and a proton, meaning H+. But our product for this step is 1,3-biphosphoglycerate. So we have a total of 3 carbons, and carbons 1 and 3 each have a phosphate group. Importantly, we have 2 molecules of 1,3-biphosphoglycerate now. That means everything from here on out has a stoichiometry of 2. As a side note, I was telling you to treat ATP as a sort of energy currency, what to keep track of in terms of how much energy we're producing. What that boils down to is when something has a lot of phosphate groups attached to it, it's storing more energy. This product has 2 inorganic phosphate groups on a total of 3 carbons, so it's holding on to a lot of energy. Step 7 is transfer of an inorganic phosphate to ADP. So we are finally producing some energy. This is catalyzed by an enzyme called phosphoglycerate kinase. It's a reversible reaction, and we are utilizing ADP, adenosine diphosphate, and we are adding an inorganic phosphate group onto it so that it becomes ATP, adenosine triphosphate. And our product is 3-phosphoglycerate. We still have 3 carbons, but now we only have 1 phosphate group and not 2. Step 8 is going to be a carbon 3 to carbon 2 inorganic phosphate transfer. So this is essentially an isomerization reaction. It's catalyzed by an enzyme called phosphoglycerate mutase. This is a reversible reaction. And our product is 2-phosphoglycerate. So we went from 3-phosphoglycerate to 2-phosphoglycerate. Step 9 is a dehydration reaction, meaning we're getting rid of some water. So this is catalyzed by enolase. It's a reversible reaction, so you're seeing 2 directional arrows. And we are also producing a water. Our product is Phosphoenolpyruvate. And last but not least, step 10, transfer of inorganic phosphate to ADP. This is catalyzed by the enzyme pyruvate kinase. It's an irreversible reaction, and we are finally making some energy. So ADP, adenosine diphosphate, is lower in energy than ATP, adenosine triphosphate. And when we break that phosphate group off of our reactant and add it to the ADP, we end up making a higher energy molecule in the form of ATP. And our product is pyruvate. At the end of glycolysis, pyruvate can have a few different fates, depending on the organism and whether or not there's oxygen. This lecture is not really about these, but any of these pathways are going to produce even more energy from the pyruvate, and they all have the added benefit of converting the coenzyme NADH back to NAD+, so that NAD+ can be used in another round of glycolysis. I have 2 questions for you. Question 1. If 1 mole of glucose goes through the glycolysis pathway, what is the net number of moles of ATP produced? You saw 2 steps that converted ATP to ADP using energy, and you saw 2 steps that converted ADP to ATP producing energy. But energy is produced overall. You should get a positive number. Question 2. In step 3 of glycolysis, which was the conversion of fructose 6-phosphate to fructose 1,6-biphosphate, the enzyme is inhibited by ATP. Explain why this is beneficial to the organism. So in step 1, glucose to glucose 6-phosphate, the enzyme is inhibited by the product, glucose 6-phosphate. This is extremely beneficial to the organism because this step is irreversible. It's a problem if we make too much glucose 6-phosphate. But in step 3, we see the opposite. ATP is a reactant that inhibits the enzyme. In other words, if there's too much reactant, the reaction slows down. This might seem counterintuitive, especially if you're remembering Le Chatelier's principle from general chemistry. So explain why the inhibition in step 2 is beneficial.